Okay, so if you are in group 2 for the Monday or the Tuesday lab, you guys will be online this week. And so this is just a sort of video announcement to help you understand what you can be doing at home to be progressing and not falling behind. Okay, so you'll see on the syllabus for 280 that you have 2 lab exams in lab. Obviously they're in lab, they're lab exams. They're each worth 5%, and this is what we're going to get started working on. So if you go to the 281 page, go to modules, you're going to see, well, let's see, if I do this in student view, you're going to see this lab exam 1, invertebrate form and function. So this is going to be the materials for the first lab exam. This is nothing. This is just where I actually input the grade. But the materials that you can use at home to be studying will be found in this module. Okay, the study guide basically is going to show you all that you need to know for this lab exam. This is some background information. Now, for each of the phylum, and this is spelled wrong. This should be porifera, phylum porifera. For the sponges, right, you should recognize these different structures and know their function. Okay, when you get to lab next week, here are the materials that you have in order to understand these, recognize them and know their function. There are some slides. Grantia is a genus of sponges, and we have cross and long section slides in the lab that you look at, and then also this page number goes along with this book. All right, so the Van de Graaff's photographic atlas. So if I go to page 31 of it, no, shoot, wrong one, this one here, whatever edition you got. Okay, so if I go to page 31, then I'm going to see this is exactly what the pictures and the underneath the microscope look like. Okay, and all of these structures that we see on our study guide are found in the book on those. So you can be looking in the book, looking at the pictures, reading up about their functions. All right, here's just a picture. This is what a grantia looks like, and here's a long section, longitudinal. Right? So that's basically take a object and slice it long ways. That's the longitudinal section through this organism. And in this slide, you should be able to see most all of these structures. And it just goes on, right? Here's a little space for you. This is basically for next week when you're in lab. It just is helpful for you to draw something as you're looking at it. Next, phylum Cnidaria. Here are the things you need to recognize and know the function. Right? Platyhelminths, Mollusca, Annelida, Nematoda, Arthropoda. This is all the material that will be on the first lab exam. Okay, and so you need to be able to find these things on a slide, or recognize them rather. I'll have it found for you, a pointer on that structure. You'll need to tell me what the name of the structure is. You'll need to tell me the function of the structure, and to be able to tell me which taxonomic group it comes from. Does it come from Porifera? Does it come from Cnidaria? Does it come from Platyhelminthes? Which class is it found in? So on and so forth. Okay, so this is all on this study guide. I would print this and keep it with you. Bring it to lab with you, certainly. Okay, so now what I'm going to do is make a series of videos, and I'll post them here under lab exam 1 that shows you each of these phyla and how to find those different structures under the microscope. And lab will actually review how to use a microscope. We'll review what the slides mean, things like that. But at home, you can be going through the study guide, watching the videos, looking at your book. Also, there's some practice pictures here. Right? You can open this up and kind of look and see. Basically, they're just additional pictures of the same things that you can see in your book. But remember, each slide is going to look a little different. So, spinning, spinning, spinning. Yeah, so here's the Grantia long session. There's Obelia hydroid. There's a Clinorchus. Here's a tapeworm. Shows you where to find the different stuff. Right. So here are just some resources for you to use to, again, in those off weeks, give you something to do. Don't wait on this. I'm telling you, if you guys wait on this, you're going to screw yourself over. You will not do on this first lab exam. First lab exam, if you spend some time on the off weeks, you come to lab in earnest, get under the scope, make yourself some flashcards, study this stuff, it is a breeze. Okay? It's not actually that much stuff. You just have to have a little discipline to study it. Okay, I'm going to sign off and post this. Look for videos of the individual phyla.